Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hello again, welcome everybody. This is our last webinar of the calendar year for the SB Grid webinar series. The next one is going to be January 10th, and that's going to be Chris Wadby talking about Titan. And if you have questions during today's seminar, please feel free to send them in the chat in chat, and we'll have time for Q&A at the end. And with that, I'd like to thank Thomas Simonson for joining us today, and he's here to present on the Proteus software for physics-based protein design. So with that, I will turn it over to you, Thomas. And once again, thank you for being here. And this looks like interesting stuff. So
thank you, Pete. Yeah, very glad to be here virtually. So Proteus and physics-based protein design. If you'll allow me, I want to just digress for a few seconds. First of all, the other day I did something slightly foolish. I added a turnip to a perfectly good recipe for leek and potato soup. And all that I accomplished was kind of flattening the taste and turning the soup purple. So it turns out today I also have to juggle with 3 flavors. I need a little bit of theory. I've got a chunk of technical information and kind of a quick view of some results. So I'm going to try to do this without going over time and without turning anything purple. I'm going to go full screen here, be a little bit clearer.
Yep,
and
full screen looks good.
So protein design basically starts from a fold. We choose a fold and we try to decorate it with sequences that have good properties. So maybe they should stabilize the fold, maybe they should bind some ligand, and probably they should do both. So we have a notion of kind of exploring a space of sequences with some kind of randomized mutagenesis, and we have a selective pressure applied. What we do, we consider to be physics-based protein design for 2 reasons. So the first thing is the energy function. So we have what we call an MMGBLAK energy function. So we've got molecular mechanics with solutes, and we've got solvent with a dielectric continuum polar part, a GB model, generalized Born model, and then a nonpolar component from Lazaridis and Karplus, an LK nonpolar solvation. That's the first thing. The other thing is when we explore the space of sequences, we make a special effort to achieve Boltzmann sampling. But why would we bother to do that? Well, first of all, that's the experimental reality. You know, proteins obey Boltzmann statistics. And the second thing is it means we can compute free energies. And possibly, or not possibly, in fact, we can actually design for free energies, which is definitely elegant and possibly important. Not completely clear yet. So in our hands, design or exploration takes the form of Monte Carlo, where moves can be mutations or rotamer changes, conformational changes. We have this vast space of sequences for most design problems. So we make a very drastic approximation, which is to make a discrete conformational space. So we typically have one fixed backbone conformation, or probably a handful, possibly a handful. And then we have a library of side chain rotamers. Okay. So the Monte Carlo moves can change side chain types, change rotamers, or some combination. This is a pretty good review right down here. With a discrete space, we can in principle pre-compute all the pair interactions. And this is old. These are old ideas going back at least to Steve Mayo's work. If we have, let's say, a protein with 3 residues here, we can set up a 2D table where along the edge here, we put the 3 amino acids, 1, 2, 3. And in my example, each amino acid can have 2 types, A or B, and each type can have 2 rotamers. So basically, we make a table where along the edge, we line up the sequence, the side chain types, and the rotamers, all the allowed values. Same thing horizontally, and this is going to serve as a lookup table. We're going to compute the interactions for between 2 side chains with some combination of types and rotamers and tabulate it. That's going to serve as a lookup table, making the Monte Carlo exploration very efficient. Now, I have one kind of point of theory I want to spend a couple minutes on, which is the solvent model. And this is very specific to Proteus, which is why I think it's worth mentioning. This lookup strategy, in principle, doesn't actually work with a continuum solvent, including a generalized Born solvent. The GB interaction between atoms takes the form that's shown here, where we have this interaction function between atoms I and J. The thing is, it depends on their separation, but it's also parameterized by so-called solvation radii here, these B values, which measure the depth of each atom. Now, clearly the depth of an atom, say atom I, doesn't just depend on J, but it depends on all the other atoms, right, which build up the protein. So the interaction term is actually a many-body term. That's not really going to allow you to use a 2D lookup table. What it means is the dielectric continuum solvent model is not pairwise additive. So for many, many years, everybody who'd used this kind of model in protein design, a lot of related problems, used an effective average dielectric boundary. Okay, just to make a long story short. So you kind of smooth the protein boundary, you forget about side chains hopping around between rotamers, you just take a kind of average protein solvent boundary, and we have that in Proteus. We call it the native environment approximation. In the software, it's referred to by a keyword NEA. Or we have a much more sophisticated procedure, which is basically exact, which uses a double lookup system. And I won't go into detail, that would be a little tedious. Proteus refers to this as exact, or FDB, for fluctuating dielectric boundary. The only point I make is that for the interaction, the GB interaction, what we do is we express it as a power series relative to the solvation radii. And these coefficients here can be tabulated. So we have a double lookup procedure where during Monte Carlo, we're going to work out the solvation radii and look up these coefficients. So we're back now to a 2D lookup table with a bit of overhead and a bit slightly increased cost. And that's, Proteus is the only tool that has the many-body GB character. We have a nonpolar solvation model. Won't say much about it. We use this Lizardis-Karplus, well-established component. Just 3 points. First of all, we also have a dispersion term, which we don't use much. Second of all, since the dispersion hasn't really helped much in our internal benchmarks, we're basically running with the GB and LK combination. That's our workhorse solvent. And the third point is that all these variants have been carefully parameterized a few years ago, very extensive benchmarking and whatnot. So Monte Carlo in sequence space, this is perhaps my last point of theory, I think. I think it's my last theory slide. What does it mean to do Monte Carlo in sequence space? Well, if we make a mutation, let's say we turn a side chain from black to gray here, what we actually do is in the folded state, we turn black from gray, and then the unfolded state, we make the reverse mutation. So effectively, a side chain mutation really amounts to unfolding one variant and refolding the other. Okay. Now, there's a simple interpretation to this Monte Carlo scheme, and one can show that the simulation actually mimics nicely an experimental system. In fact, it mimics an equimolar collection of sequences at equilibrium and distributed between the folded and unfolded states according to Boltzmann statistics. So that's the interpretation of the simulation scheme. And we don't usually do plain Monte Carlo. We usually do replica exchange Monte Carlo. So we've got a bunch of replicas, 4, 8, maybe as many as 16. And this is a time course, a simulation course here, where you've got 8 replicas sort of at these different temperatures, which are in KT units. And occasionally they swap conformations, or alternatively, they swap temperatures. So you can see a replica going up, down, up and down the temperature line. And this is very efficient with if you make the effort to install OpenMP on your desktop machines. Now, before going into the kind of technical details, a little bit of technical details, I just want to point out that with this energy function, we have been able to redesign completely a PDZ domain. And we had a handful of designs that were tested, and in the latest batch of calculations with the best model, the three tested sequences did indeed fold. Now, the number of redesigns here is definitely underwhelming, because nowadays people are doing these high throughput designs and experimental validation. They've got these robots going like crazy, and they're doing hundreds or even thousands of redesigns they're able to test with reasonable hit rates. But you can't get a better hit rate than 3 out of 3. So although the number is low, this was the first time a physics-based energy function was able to produce a successful redesign. Not because it's tremendously hard, because people simply a lot of people moved away to the knowledge-based function. Okay, so the Proteus software, the current version, up to maybe a trailing digit here, was released in 19. These are the authors. So Proteus is very much part of a family of programs that started with CHARM, went to Explore, Axel Brunner at Yale, CNS with a bunch of people. I was a developer as well. And what these programs and also Proteus have a couple of things in common. They share a lot of their logic. One thing is they each have their own scripting language. And that's actually, it works actually maybe surprisingly well, surprising maybe for younger people in the audience. And they share a lot of data structures. And hopefully some of you are familiar with those data structures, because I'm not really going to spend a lot of time talking about the data structures that come from this molecular mechanics world. I'm going to spend a bit more time on some of the more specific features of Proteus. So on the website, you've got a detailed manual. You've got a large set of tutorials with both the fully worked version and the version that's ready to go. It's downloadable, and there's contact information and some other stuff. So a few other resources. Again, the manual is this old-fashioned concept of a PDF file. It's still searchable and what have you. Now, because of the close kind of phylogenetic relationship with Explore, turns out the command language we simply took from Explore. And we've got a lot of functionality related to energy, subroutines, and so on. So the Explore manual is also a useful resource, and that's freely available online. We've got an email hotline, and really feel free to email us at any time. You can also email me directly. And there are a lot of publications. There are a few chapters in this kind of how-to series. These books, they're sort of how-to chapters, and we've got a couple of those. I have some articles I list at the end, and then there are the tutorials. And we've got this is a pretty big set of tutorials, which are, again, quite detailed. So we've got plain design of a very small system, a peptide, plain design of a PDZ domain. And you've got this important Proteus feature, which you can design for binding with adaptive Monte Carlo. And there are a few other groups that do that with kind of coming more from the computer science side with methods that are a lot more expensive. They don't have our throughput. And that's provable based on publications. We've got an acid-base tutorial. We can do multi-backbone design. And there's a kind of a complicated problem here, which is pretty well described, optimizing parameters for the unfolded state. That's for whole protein design. So there's some resources out there. But especially, we're online, we're available. So looking a little bit more inside the Proteus box, there are really two main pieces of code. There's the Prodex code, which is the one that's the closely related to Explore, and which handles basically energy calculations. Remember, the energy matrix is calculated ahead of time as a lookup table. So it's a separate program doing that. And ProtMC is our Monte Carlo code. So this guy is mostly Fortran 90 with a little bit of C, and this is entirely written in C. There's a lot of macros written in this Explore CNS script language. These are like little macros, thousands of lines of them. And then there's a Bash, Perl, and Python layers that are usually up above these layers here. And Bash, we really expect this to run in a Linux environment. We have not made the effort to, try it out on Apple Unix, for example. We haven't really felt the need to do that. And if you like Windows, make a virtual Linux machine on your Windows box, and we will happily distribute. We can give you a virtual Linux machine with Proteus installed, actually, or just use SPGrid. Right? So this is too short a format to go through any tutorials in detail. What I want to do is give a little bit of flavor. Now, you will see through these, going down kind of to this more technical level, you might perceive a little bit of clunkiness, but it turns out the clunkiness, there's a trade-off, and all of this is actually extremely tunable and controllable and powerful. So it's a bit of a trade-off. We program things at a kind of low level, and frees up a little more time to do theory and things like that. So you're basically starting out from a system with a PDB structure, and then you have a kind of a build step. And this is build would be understood by users of Charm, Explore, CNS, NAMD as well. This is all very compatible with NAMD. And it's basically making a PSF, which is like a 2D description of your molecule. It's like a 2D drawing of a giant protein, which gives you a lot of logical information, such as where are the covalent bonds. So you build up a modified PDB file, which has hydrogens, atoms, and a few other things. It's PSF file. Then you go into a setup phase where you've picked a few positions you're going to allow to mutate. Could be the whole protein, or it could be a few positions in an enzyme active site. Okay, I'll give an example later. What we do for these mutating positions, we put all the side chain types there with all their rotamers, something we refer to as giant residues. Okay, and you've got a couple of files here where you can control which residues will mutate. We call them active. The others are inactive. And some parameters of the model. There's really just one force field that's supported, that's well supported, though in theory, there are a couple others that are partly supported. And if you have ligands, they would have their own PSF file, for example. Then you go through a bit more setup, and this includes things like putting rotamers in place and computing GB solvation radii on a residue atom basis or residue basis or both. And then computing the diagonal part of that energy matrix. Okay, and over here, you compute the off-diagonal matrix, energy matrix elements. And these are controlled by these Prodex scripts, which use the Explorer script language. This one, these ones here. There are also a couple up here. Now you're ready to run Monte Carlo simulation. There's a bash script that will, do that for you. And you're going to get sequences in either a raw format or more human readable format, which I'll show a little bit more. This is the same thing in perhaps a little bit more linear, slightly more detail. You've got a project directory, and then down below, you've got a couple of different tasks that have their own directory, a build directory, a matrix directory, some macros in here. Your personal parameter choices would be in these macros. And then you basically have shell scripts that control the different calculations. And at this point here, you have an opportunity to use the GNU parallel tool to parallelize some of this. And then the PRODMC, the Monte Carlo calculations, again, those are parallelized within the bash. You don't see it here on the command line. A quick look at what this energy matrix looks like. So the diagonal part, you basically have an amino acid position, and you have one line here for a particular type and a particular rotamer. Okay, so this amino acid position could have 10 or 100 or 200 lines associated with it, going through the different types and the different rotamers. You've got different energy components, including things like van der Waals, Coulomb electrostatics. Over here, you've got some GB information. This information allows you to run this NEA approximate GB. And then there's some extra information here and over here, which allow you to use this interpolating function and this double lookup procedure. And then you have analogous information on the for the off diagonal. Now the complexity has been, raised to the power of 2. So you've got 2 amino acid positions, each with a particular type and a whole bunch of rotamer possibilities. So these are human readable ASCII files. They can be edited by scripts in a lot of different ways. Okay, you can change the protein dielectric constant by editing these files with a Perl script or a Python script in a well-automated way. Not something you'd have to develop yourself. The Monte Carlo is controlled by its own command file, what we call a conf file. So this one is called mc.conf, and it has an XML syntax. And this is a pretty good example. It shows how to run just a plain Monte Carlo simulation. So you've got a Monte Carlo task. You've got the location of your energy matrix lookup files. You've got a 10 million trajectory length, which is very short. We typically run more like a billion or billions of steps. You've got some numbers that help control the calculation by introducing some interaction cutoffs or making things more efficient in a couple of different ways. I've only left this one as a kind of illustration. This example runs with 4 replicas. These are the temperatures, KT units. This is room temperature up here. Replica swaps every 2,000 steps. 1, 2, 3, 4, 5 types of Monte Carlo moves, single position or dual position. GME method here is the exact, and this could also be FDB would be a synonym. Proteus can use either one. This is a raw sequence output. This is an energy output file. It's partly redundant. And then you have parameters for the unfolded state. Remember, we're always propagating both the folded and the unfolded state. And the unfolded state is a very simple model with just one parameter per amino acid type. This is very typical in protein design, and they can be provided here. And this particular file would run a Monte Carlo simulation, which would have 4 replicas, not 8, and so on. It would look something like this. Just a very quick look at some of the main output files. And then I think I'll move on to a little bit more an application flavor. So this is the raw sequence output, and each replica has its own set of output files. So this is a file associated with replica 0. It's the lowest temperature replica. And these are just the first few states that were sampled in the trajectory. The file goes on and on and on. And if you run 1 billion steps, this can be fairly big. So these would be files you would compress rapidly or trim rapidly, and just filter based on energy, all sorts of things you can do with these files, but they start their life out as ASCII files. You've got an identifier for the Monte Carlo step, got a residence time, how long the simulation or this particular replica spent in the corresponding conformation. You got an instantaneous energy for this particular replica, and then you got rotamers, and this is an internal numbering scheme for the rotamers. Then Proteus can write out this rich format, convert it to a rich format, which is more human readable. And now the residue numbers, the amino acid types are explicitly given, whereas otherwise they're hidden inside the rotamer numbers. And the rotamer numbers here are local to each position, where these rotamer numbers run over the whole system. And when you run the Monte Carlo with BASH, usually the BASH will finish up by doing a little bit of very basic analysis. And one thing it will produce is a bit of statistics on the most populated sequences for that replica during the simulation. So for example, here, in my example, I just 2 positions mutating. This is a toy system, mind you. And so you've got the top sequences. This is the percent population. And these are the amino acid types. The average energy for this particular sequence, and then the interval of energy. So, just a little bit of basic statistics right off, just immediately output. Okay, so now I want to spend a little bit of time, a few slides on what is really an important example. And again, this is something that's very specific to Proteus. So first of all, our main interest is enzyme redesign. Okay, that's the kind of application we're interested in. Everything else is testing and benchmarking and, very important stuff, but the actual applications we're really interested in is enzyme redesign. So what does that mean? It means you choose a few amino acid sites in the active site, and you explore mutations in search of activity defined in some way. So your design criteria, of course, you need to maintain the stability of the enzyme. You don't want to unfold it. And then you could have a criterion that could be substrate bounding, ground state binding, or it could be transition state binding, or it could be some kind of specificity criteria, like bind the new substrate, not the old substrate, things like that. There are a lot of variations one can make. This is a hard problem because you need, if you want to design for substrate binding, you need positive design for the complex, the holo state, and negative design for the apo state. That's actually hard to do in a simple way. The methods that do achieve it are all pretty complicated. Now, the best one by far uses so-called Wang-Landau adaptive Monte Carlo. So this is a rigorous method to sample by affinity. Don't worry, I only have a couple of slides, on the methodology. This one and maybe one more. The idea is incredibly simple and elegant. First of all, one adaptively flattens the free energy landscape of the apo state. Okay, so you do that by learning a bias function, which you add into your energy in such a way that the sequences all have comparable probabilities, meaning that you flatten their free energies. They're all at the same level. Having done that, your bias function is approximately the free energy with the sign change. Something to mull over, maybe, later over coffee, you can mull this over, or you can, look at the articles. Now, if we simulate the holo state with the same bias, the bias subtracts out the apo free energy. It's really amazing. It's like magic. It's very easy to understand, spend a few minutes thinking about it. So this is a simple example where we have a very complex enzyme, but we're only mutating 3 positions. And we're mutating 3 positions partly because this is an illustration, partly because this is a hard problem. We can't flatten the free energy surface if the dimensionality gets to be too big. So 4 or 5 positions, fine, but 50, no, we're not going to be able to flatten that big a space. This is a sequence logo for our 3 positions in the apo state without any bias. These are the natural probabilities of the different types at each of these 3 positions. Now we go through this adaptive learning procedure, and we reach a point where all the sequences have very similar probabilities. And so the logo just looks like a mix of everything. And now we add the ligand to form the holo state. We keep our bias in the simulation. And now we get a logo where the sequences are populated, believe it or not, according to their binding free energies, which is really what you want. Okay, so if we make a logo where the populations are weighted by the Boltzmann factor, the exponential of the binding free energy, this is the logo. And interestingly, the native sequence is LAI in this particular example. This is not a toy example, it's a real example. And this is actually the native sequence. So this is actually, there are only 2 tools that do this. One is Proteus, and the other is Charm, with Charlie Brooks's work. Charlie's work is great, but it's a lot more, the model is much more expensive and doesn't have the same throughput. So they're complementary tools, actually. These are the only 2 pieces of software that can apply this adaptive method. So there's a tutorial that covers this in detail. Just a few kind of salient features. First of all, the tutorial uses the tyrosine tRNA enzyme. Sorry, the tyrosine tRNA synthetase enzyme. Dropped a word there. There are 5 active positions, and you can see this. This is a little piece of a conf file. So when you run the Monte Carlo, Proteus is very good about allowing you to control the mutation space. When you start out, you pick maybe 5 positions that are going to be able to mutate. But when you go into the Monte Carlo level, you can reduce it in a lot of very flexible ways. So in this particular case, each of these positions is only allowed to have its native type. These are the native types, or the alanine type. This is now a little bit of a toy problem for the tutorial, okay? That's one point. It's a tyrosyl synthetase, and it's got 5 active positions, and you have this kind of just one additional level where you can control the mutation. Now, we've got a ligand. The ligand here is azidophenylalanine, and you can see it over here. It's a non-natural amino acid, which has been incorporated into the genetic code by engineering this enzyme. For any problem with Proteus, where you want to design a system that includes any ligand that's not an amino acid, okay, so an unnatural amino acid or whatever, small molecule that's of interest to you, you always need to provide topology, parameters, and rotamers. Now, rotamers are the simplest because for Proteus, ligand rotamers are just poses. So you simply come to Proteus with bringing your set of poses, each in its own PDB file, or grouped in a single PDB file, also fine. Okay, so ligand rotamers are just poses, and it's your job to bring those poses to Proteus. Proteus will not produce poses or rotamers for your ligand. That's upfront. You run AutoDA, AutoDA, whatever. Topology and parameters, this is now kind of charm speak, and hopefully you know what this means. So here you see I'm reading in topology files, standard files for protein, and here I've got a file, custom-made file we built in-house for this ligand for a few years ago, not for a toy study, but for a real study. And parameters, we developed our own force field parameters for this ligand. It's not very hard because there's only this tip, which is new. Everything else is just like phenylalanine. This, from ligands, you need to come with bring your parameters. Okay, those could be GAF parameters, they could be CGen FF parameters, they could be lots of things, but Proteus will not parameterize your ligand. And then the main steps of the tutorial are listed in the manual. You build the system. Build has the meaning I described earlier. It's charm speak or explore speak. Build the system in the apo and holo states. Pre-compute the energy matrix for each one. Compute unfolded energies. This is very well automated, don't worry. This is pretty much push button. It's not a big deal. Adaptively learn the optimal bias for the apo state, and I'm going to come back in either just one more slide to help with that. Compute the biased populations of sequences. So this is a kind of beefed up version of the earlier analysis I mentioned. This is well automated. And then sample the holo state using the same bias. Okay, so Proteus will write out the bias function it has learned, and then you will read that back in to simulate the holo state. So just one last slide to speak a little bit more about this whole adaptive bias, because it really is an extremely useful tool. The idea of the bias function comes from Wang Landau back in 2001, but it's also very closely related to metadynamics. Now, I don't know if anybody in the room knows about metadynamics. The idea is that every so often, every n Monte Carlo steps, we look at a position we want to mutate, say position I, we look at what the type it occupies, and then we put into the bias a little increment that penalizes that type. So the bias is constantly trying to penalize the types that it's seeing in the simulation. And what that does is if you sample a particular type over time, you sample it a lot, after a while, you want to move away and sample other things. If you want a flat surface, this means you're sampling everything. So you want to penalize the stuff that's being sampled, and eventually other things will be sampled. And eventually you'll be sampling everything pretty much at the same level. And the trick is that these little increments, they get smaller and smaller. And that's been worked out by the metadynamics people in great mathematical detail. And so you have this form here for this little increment, the bias energy for position I, for a particular side chain type S at a particular point along the trajectory. And this little increment, it's getting smaller and smaller. It's decreasing exponentially with time. So that's kind of what the bias looks like. Now, seen from Proteus, it's all pretty much automatic. So in your conf file, now I'm calling it adapt.conf. because we're going to be running in Monte Carlo to learn, adaptively learn the bias. So here I'm telling Proteus what are the positions I want to bias. All 5 of my positions that mutate, I bias all of them and flatten all of them. And I'm going to give Proteus a few kind of technical parameters, like how often, do I increment the bias? How often do I write it to a file? And what is the output file for the bias? So like here, every 10,000 steps, Proteus is going to write down the current bias. And that could be very interesting. Over a long time, you get convergence, and that's the file you really want to keep. So that's basically just running the protMC binary with this adapt.conf file. And one of the things it outputs is the bias all along the trajectory, the sequences that are visited. And at the end of the adaptive procedure, when we feel that everything has converged sufficiently, we take the final bias and we copy it into an input file. And that will be used now for future sampling. Okay, so these are just things that get added into the basic Monte Carlo command file. Just mentioning in passing this one application system. This is a methionine tRNA synthetase system. We designed for this non-natural amino acid, and we recovered the top experimental hits, which were already known. We redesigned for methionine activity, which is the natural substrate, and we predicted a whole bunch of hits that should be active. And subsequently, they were tested, and they were indeed active. None of them was more active than native, but they were all active. All the tested variants were indeed active. There were 19. And more recently, we designed for a bona fide application activity towards beta methionine. And there too, we had some nice hits. And the important thing I want to mention about this application, this was designing for transition state binding. This is an example where this was a simple model of the transition state for the synthetase reaction. We designed for transition state binding, not ground state binding. So that in fact, the adaptive landscape flattening allows the design of an enzyme either for binding or for catalytic power, literally. So that's elegant and possibly important. These are some slightly more important articles. It's not a complete list, obviously, but there are a few down here. This is the original adaptive landscape method applied to protein design back in 2018. This was our successful PDZ redesign here. This is the enzyme redesign I just described. This is a review I already mentioned. And these are both how-tos for PDZ peptide binding. That's a nice example of protein ligand binding. And then this is a more complicated problem, which is parameterizing the unfolded state model. Okay, that doesn't come into the enzyme redesign at all. Nice thing about the enzyme redesign with the adaptive landscape flattening doesn't depend on the unfolded state model, or very, very weakly. Okay, and just to wrap up, these are the actual Proteus authors, and these are other people in the group who have contributed in other ways. They've done applications, they've done some benchmarking, they just haven't been really code developers. And down here, I have collaborators only on the theory side. I haven't listed experimental collaborators, it's not the nature of the talk. Very long, fruitful collaboration with my very old friend, George Arcontis, who's in the physics department in Cyprus, some colleagues nearby in Paris, these guys, Carlos Nito, also in Paris. some computer science folks in Toulouse in the south of France. And I also acknowledge Axel Brugger because he very kindly allowed us to use code and also documentation taken directly from Explore. So with that, I'll thank you all for your attention. And I guess we have a little time for questions. I haven't really checked the chat.
The chat I've been watching, and it's been very quiet. So
Okay.
there's no questions in the chat so far. If you do have questions, please feel free to send them, or you can use your raise hand icon and we can unmute folks or let you unmute yourselves and go from there. And I have a few questions that came up as you talked, and you were talking about dividing things up into 3 sections, and I think kind of accidentally my questions land in all 3 of them.
Okay.
So on the theory side, for the lookup tables that you mentioned, does that account for the distance of the interactions, or is that just thresholding?
Yes, no, that accounts for the distance in the interaction. So for example, for the GB here, you see the interaction between atoms depends on their distance, but it's parameterized by their burial. So Rij is the distance, and Bij is the average burial. It's the average of these 2 numbers. So it's parameterized by the burial, and so you have different regimes. If you're looking at an exposed pair, then the interaction looks a lot like just plain Coulomb. It looks green. If you're looking at a very buried pair, then you have different limiting behavior and your interaction looks blue. It's actually very small. Why? Because it's a solvent contribution. If you're buried, solvent doesn't come into it. And so you're blue, you're down here, it's very small. So there's this double, there's this distance dependency, but parameterized by burial. And when you go to this method here, we're formally writing the B dependence in an explicit way, but the structural, the structure for the pair Ij of residues, it's included in these coefficients, which are actually huge lookup tables. Each one of these is a 2x2 table.
Yep.
So.
Thank you. All right, and no more questions are coming in, so I'll keep going down my list. For people that aren't as familiar with Monte Carlo sampling, I guess the first question is kind of how sensitive is the process to the optimization parameters? And then that was a kind of generalization about, do you have any rules of thumb or guidelines for number of steps versus number of replicas versus how frequently exchange happens?
Okay, yeah, thanks, Pete. So actually, one thing I completely forgot to add to my slides was any kind of timing information. Sorry about that. The study of an enzyme redesign with 4 or 5 mutating positions, that's basically 1 to 2 days on a single desktop machine.
So that's pretty fast.
If you've got 16 cores or upward of 16 cores, so an inexpensive desktop machine, this is the work that we used to run this with a PBS batch queue on a cluster. And over the years, we just stopped, everybody just runs their calculation on their desktop machine. And then what eats up cycles is when we run these designs through an MD simulation with explicit solvent. That's the second thing I forgot to mention. We set up, Explore and MD have a close historical relationship. And our build process is compatible with NAMD. So these guys can go directly into NAMD for explicit solvent MD. And that can be a lot more expensive. So this is moderately expensive. It's not, these calculations are not very expensive. In terms of simulation lengths, how long do you need to converge a bias for, say, 4 positions? Definitely hours. the exact GB procedure is about 4 times as expensive as the simplified. It's not an order of magnitude. And you might need to run calculations for maybe 2 days to flatten 4 positions with the high level GB, and then sample with the bias. In practice, we commonly will run, 1 billion steps is kind of a solid length for a production simulation. So that's one kind of short answer. In terms of initial guessing and things like that, there are a couple of pointers in the manual. One thing that helps is to start off with some reasonably good initial guesses, and those can be taken right out of the energy matrix. It's not very hard to look at the energy matrix and come up with a rough first guess of a bias function. And that's well automated as well. It's well automated. But convergence, if you run billions of steps, things will converge. Sometimes you need to intervene manually. There are times when something is so far off, it's never going to converge. So you need to kind of supervise the simulation. It's not completely unsupervised. So from time to time, you have to step in and just manually change a bias value. There's a little bit of experience that's needed, but it's not super duper rocket science to see that, that's happening. You might need a little bit of help. It's not very hard.
Okay. Because I was curious about that because I've run across cases where, large optimization problems, and sometimes the parameters you use, they just say don't matter, let it run longer. Sometimes it's if you don't get them exactly right, everything fails. And it sounds like this is somewhere in the middle of that.
Yes, you need to supervise things, and there are cases where you just have to step in and throw out an amino acid type that's never going to be brought to the same level as the others. There's simply not room. In theory, you can patent things, but if there's no room, you got a fixed backbone, you could need a vast bias energy. So you need a bit of supervision.
All right. Well, thank you. And we have a question from James Wachira.
Yeah. Thank you so much. I have a slightly different question. And so the problem we are dealing with is having 2 enzymes that have got related sequences and they work with related substrates, but not exactly the same. Is there a way to use this program to find out what the effects of those changes are? In other words, to do a comparison in terms of what has changed moving from one enzyme to use to one substrate and the related one to use a slightly related substrate?
Well, I'm pretty sure there are things that can be done, but I would rephrase the question kind of closer to the typical protein design question, because the typical protein design question would be, I've got an enzyme that operates on tyrosine, and I wanted to operate on, acetamide or some analog, right? I want to repurpose the enzyme to operate on a slightly different molecule. And so, of course, that's related to your question, because you've got an enzyme that works fine for your native substrate, and then possibly you have a second enzyme that works for the second substrate, but what protein design would kind of likes best is modifying one enzyme to accept the other substrate. So that's a slightly different question, obviously. It could be used to kind of help answer your question. What are the key residues? For example, if I take enzyme A and I try to make it work with substrate B, I can mutate the active site until I get some level of activity. And that will certainly tell me what are the operating positions. right?
Yeah.
And by the way, we can do more than 4 or 5 positions because we can just do 4 or 5 at a time.
Mhm.
But you can cover the whole active site 4 by 4.
Yeah.
If you got 20 active site positions, covering it 4 by 4 doesn't mean you do 5 times 4 positions. Maybe you'll do 50 times or 100 times 4 positions. In other words, you'll cover it by 4 positions in as many ways as you want. As long as it's a few dozen, it's perfectly fine. It's perfectly manageable. So that's how we cover like a whole enzyme active site. We focus maybe on 20 positions, and we redesign a few at a time. We've had applications where we tried to do a specificity swap. So we have, say, a synthetase that likes one amino acid, and we try to modify it to prefer a different one. So in effect, we'd be scrambling the genetic code. And actually, that work also involves a lot of sequence analysis, because sequence analysis is another way to pick out the discriminating residues, right? But protein design can be complementary. It can help address that question. What are the key residues that make A what it is and B what it is? I'm not sure it can formulate that correct question exactly directly.
Yeah.
But it can help.
Yeah. Okay. Thank you so much. Very interesting work.
Thank you.
One other question, since we've still got a few minutes. So you'd mentioned with the flattening and targeting the transition state as a possibility versus the pre-transition state.
Right.
Is this a model that you could extend to include like the ligand and the bound conformation and the transition state and the product state as well as the apo state? Or and I apologize, that question may have been incoherent. So if it was, please let me know.
We can establish a competition between any two of those states. So what I said for the establishing a competition between apo and holo with just a single ligand, there's a slightly different way to do it from what I described. So the way to do it is to aggressively sample apo. It means you flatten the mutation space. Now you've sampled everything in a controlled way with a known bias. You can back out the unbiased populations. Do the same for the holo with one ligand, say the reactant. Do the same thing now with the product. Now we can back out unbiased populations of a particular sequence in the apo state and the reactant state and the product state. The only condition to do that is that our sampling was aggressive enough that we hit that sequence in all 3 states. But the thing is, our sampling is so aggressive because we're flattening the surface.
Yeah.
That there are a lot of sequences that we will indeed sample in all states. And so we can set up a competition between more than 2 states. Just involves a bit more simulation and a bit more of this aggressive adaptive sampling. And by the way, what I showed in the kind of simple tutorial example was a bias function which was defined by single positions. The bias acted on each position I independently of the others. But we can also bias pairs of positions. And that's a more aggressive flattening because now we have a bias function that's richer, has more terms, and it's actually richer, and it gives us a better flattening in the harder cases.
Okay. So I guess if paraphrasing your answer slightly, you'd break it down into three separate problems versus one like three objective problem.
Yeah. And one of those problems could be transition state binding. And another problem could be binding of an alternate ligand.
All kinds of flexibility.
Exactly. So,
Cool. Well, thank you. And I guess one last call for questions, even though we're kind of at time. Going once, twice. Well.
Okay, well,
Yeah.
thanks for folks for being here, and it's been fun.
Well, Thomas, thank you for presenting. It's been really interesting.
Okay, have a nice afternoon.
Have a good rest of your day.